Good morning and welcome to the MPG primer for this morning. We are very happy today to continue on with the project management series that we started a couple weeks ago now before the Thanksgiving holiday. Today we are very fortunate to have a talk by Dr. Namrata Gupta, who's the Director of Scientific Partnerships and Project Management at Broad. She earned her PhD in pharmacology and therapeutics from McGill, and as a postdoc, she studied stem cell biology and the genomics of Huntington's disease. Namrata joined Broad and MPG in 2010, initially managing the HIV Gates Initiative. And today she leads a team of 14 Broadies who manage projects and partnerships supporting platforms and programs, including MPG, the Cancer Program, the Data Sciences Platform, and the Genomics Platform. And throughout her work, she has contributed to shaping the tools that are currently used by Broadies who work on project management. In 2018, she also earned her PMP as part of the Broad-initiated PMAP program. And a couple notes on logistics for today. Namrata has kindly agreed to answer questions that arise through the talk. So Sarah will be posting some directions on how to raise those questions, and I will do my best to interject them at the appropriate time during the talk. So please feel free to raise questions either then or at the conclusion of the talk. So thanks again, Namrata, for agreeing to speak today about the A to Z of project management.
Great. Thank you, Dan, and thank you very much for hosting this. And as I already mentioned to you, I'm pretty excited about this mini series on project management in general, because I think it's a very big component of the Broad and how we get projects done. So so really excited to be part of that and to kind of help people sort of navigate this at the Broad. So I thought I'd and I have to use the panel. Give me 1 second. I thought I'd start off with just reminding everybody about the really great groundwork that Chris laid for us in the first presentation in this series, where he basically talked to us about how to really organize sort of large scale multi-center collaborations, how to think about sample size to really scale up GWAS experiments to maximize power, thinking about your study design based on your goals of your research, whether it's a dichotomous disease or versus sort of a qualitative trait, the architecture the genetic disorder, rare versus complex, which then lends to designing different strategies for your study, and what platforms you would use to generate data, exomes and arrays versus sort of exomes and sequencing the genomes and exomes versus RNA-seq, things like that. You also touched base on a lot of interesting components of organizing a consortium and specific rules of engagement, and how it's really good to set these things up front to make sure that there are no surprises and people kind of understand responsibilities and stuff. And then a great list of various pitfalls that arise in projects, which it can happen because it's life and things happen. We're living in COVID world, right? So we know this too well. So I thought that I'd kind of lay that groundwork, and so based on what Chris talked about, once you sort of know what you want to do, let's focus on like how to get it done. So essentially, I am basically leading a team of project managers at the Broad and basically manage projects and people and initiatives. And the one thing that, something that I'm showing you here is that's been adapted from, PMI.org. There are really sort of key 5 phases to project management. And that's, I believe, is really your solution to basically, getting anything done, especially at the Broad. And essentially it's, as laid out here, there's the initiation, the planning, the execution, monitoring of the whole project, and sort of a closeout. And something what's important is that it is iterative, right? So, there's no project that kind of flows in this kind of cyclical way with absolutely no issues. And so it's very important to, as your project is being executed, you're monitoring the cost, you're making sure that, what you laid out in terms of the groundwork is being met, things like that, keeping an eye on the timeline and all that. So it's very iterative, but it's very important to keep this in mind in terms of, how to sort of set things up from the get-go. So the first thing that, one must do, right, before starting any project is really what, in sort of the project management world, we call a project charter. And, these are some things that I list, but there are even more things that one has to consider. But essentially, really chart out like what are your goals of the project, which should not really change unless there's like a drastic change for various reasons, but that should be kind of the pie in the sky, right? Like what is the ultimate goal of the project, so that we can always go back and refer to that, when there are deviations, and then make sure that we're still meeting those goals. Who are going to be the stakeholders in this? Who are people who are going to be responsible for different things and need to know what's going on? Where's the money going to come from? What is going to be our budget? Who's funding us? That makes a lot of differences in terms of how the project is run. Something that I like to put on the top of the list is really defining risk. I think that's really important in the kind of work we do, is that nothing is foolproof, and I think that we have to take a certain amount of risks and for various reasons. And I think that making sure we define them and agree on them is a way to sort of make sure that we can mitigate them when they do show up. Establishing timelines and milestones and making sure that we kind of hold ourselves accountable for that. As Chris also mentioned previously, this is all about collaboration, right? And so there's a lot of parties involved, making sure that there's establishment up front in terms of who needs to be informed and of what exactly, and what the cadence of that should be. And then, obviously, there's a lot of people involved, right, to get the work done, to making sure that people know in advance, what they'll be responsible for in the next few years, and make sure you have buy-in from them, to really have sort of that dedication towards the project. And then, of course, lastly, in terms of meetings, there's obviously a slew of meetings that happen within any project, and just to make sure that that is also an expectation that is laid out clearly and agreed upon. And I really state that all of these things and others really should be agreed upon sort of by all the stakeholders in the project. So what I thought I'd do is just kind of run you through with you sort of a typical project life cycle, for a genomics project that we do many of these at the Broad. And then following that, I'll focus a bit specifically on some of these aspects. But essentially, if I start from the top here, and again, it's not in a particular order, but generally, when you first start a project, you think about compliance, you think about agreements that need to be in place, making sure that consents are in order. Do you have approval to do this kind of research, etc. So that's kind of the first piece. The next is obviously funding, right? Like, what kind of money do you need to do this kind of project? Making sure you're setting up the budget accordingly once you have the funding in place, via whatever mechanism you have achieved to get the funding. We set up various quotes, within the Broad, whether they're internal work or external work, making sure that people have committed to the funds, essentially. As you know, a lot of our work is done on the cloud in terms of data generation and also analyses now. So making sure you're setting up all of those sort of Google Cloud billing accounts, things like that. And also, what's noted here is that, we really also is an important part of sort of project management, is to really help in providing, budgets and sort of text for your grants, to make sure that, we kind of lay out the groundwork for you to figure, to get your research dreams done. And then, again, focusing kind of on a genomics project, obviously, once these are kind of in place, you want to sort of execution part, you want to kind of get going, where you want to bring in samples, and we have a whole mechanism of how we do that. I'm not going to go through that in detail today, but we obviously really want to make sure that everything is kind of logged, and so we have like a really robust LIMS system available at the Broad, where everything is barcoded, everything is named a certain way, making sure that patients are linked accurately when we have like multiple samples from us, an individual patient, and obviously bringing in all the phenotype data, all the metadata in, and because without that, really can't do that much analysis. And so a lot of this piece up front is really, really critical to do well and, meticulously. A lot of our, data production flows through the genomics platform. And so, the next step is really working with the platform to, get the data generated, data review, making sure that the quality is looking good, keeping an eye on that, keeping an eye on the timelines for that. I mentioned sample swaps here because, unfortunately it happens where, many projects, we get samples coming in expecting a certain sample, but it's another sample and stuff. So this happens, it happens a little bit more often in the cancer world, where we have sort of paired patients with cancer tumor normals coming in from like different pathology labs. And so sometimes it's a bit more frequent there compared to germline projects. But it is something that we really have to dig into. And again, really making sure that we are, we know the identity of the person in the sense that, not who they are, but making sure that when we give back data, that we are giving back data for the individual that, was collected. And then, the last two steps here are, of course, delivering the data back, back to the collaborator, back to consortia, back to people who are going to do the analysis. And so a lot of that is like, as you can imagine, making sure that, as project managers, we are sort of custodians of the data, knowing what the agreements are in place, who is supposed to get the data, who is not supposed to get the data, et cetera. And so it's a very big component of that, and making sure that that's done, adhere to certain standards. And then finally, again, depending on your funding sources and kind of the goals of the project, most projects these days require, the data to be available, publicly. When I say publicly, obviously in a controlled fashion, controlled access fashion. So basically what this means is that, you have to register your study, register the consents, and I'll talk a little bit more about that a bit later, but, essentially making the data available to the public for approved researchers so that they can, further use the data downstream for other projects. So let's start at the very top. If there's any questions at this point, I'm happy to answer them, but I was thinking we'd start from the top, sort of the compliance and agreements stuff at the get-go, unless there's any questions right now about the whole cycle. All right, so let's continue on. So agreements, as you know, and as Chris had laid out, projects are done with multiple people involved, right? There are collaborations from different centers, different funding agencies, et cetera. And based on, what the project is, you may need a material transfer agreement, which, you're actually transferring material in terms of like samples. It could be a data transfer agreement because you're actually transferring data versus samples. Sometimes it's less collaborative, but more like a service agreement. Sometimes, aside from contracts, you could have, there's grant mechanisms, contract mechanisms, all of these things, depending on the nature of the project, you'll be basically looking into and signing off on. And I just wanted to kind of lay out a few things that usually kind of go into these things, right? It's these kind of agreements and contracts, they really establish boundaries, right? They establish, who is responsible for what, and, up to which point, does this sort of collaboration go till. Responsibilities, which entity is responsible to do what. Again, as I mentioned about custodianship, really important about, who really are the custodians, of the samples, of the data once it's once it's going to be generated. This is a place where, PIs can really lay out the ground rules on publications as well, in terms of, potentially even authorship, or even like, at least maybe not down to the authorship in detail necessarily, but, just in terms of, what will be the policies, and, what will, which groups will be involved in the publication. And then also, data usage and dissemination. There are sometimes there are some projects where they're very strict about, that we're going to be doing this project, and then the data goes back to the collaborator, and, they will kind of take care of the downstream in terms of who is allowed to use it and things like that. And sometimes these agreements will state those kind of regulations. Something I want to put together, in terms of sort of broad specific aspects of these agreements in general. As you know, we are obviously nonprofit, so we're tax exempt. And one important thing is that because of that, we we really want, like our legal office really wants to make sure that Broad can maintain sort of academic freedom. So we really, don't go through the route of assigning any intellectual property. That is something that we do not agree to. So that's one important part. And then another important part, as you know, is that, we're obviously, working with a ton of partner institutions, as you all know, and most of you come from our partner institutions, that, if you do agreement at the Broad, that does not equate to an agreement at your partner institution. You do have to have one separately. So for example, if you have a material transfer agreement to bring in samples at the Broad and do analysis of the Broad, you cannot do that at your partner institution. We can, of course, create through agreements and things like that for you to enable that, but just to keep that in mind before moving that forward. Excuse me. And lastly, just wanted to let everybody know that there's a website, a Converge software that we now actually use to initiate agreements and keep them all sort of organized in that fashion, so which is searchable as well, which is really nice, something that we've done more recently. So a really nice catalog of all of our agreements. And then we do have a specific, which we term OSAP, Office of Strategic Alliance and Partnership, which really help us, execute all these agreements. Moving on to ethics and IRBs, as I mentioned, obviously, all of our research that we do, requires some form of IRB approval. And there are many types of protocols that you may want to, basically execute depending on the type of your research. Without going into too much detail, excuse me, there are basically sort of 4 main categories, and there's some key differences. Essentially, your project may be deemed as sort of non-human subjects research, and what that really means is that there's really, obviously there's no interaction with the participants, and you'll get no access to any identifiable information. And one of the key components of this type of protocol is that there will be no joint publication and there'll be no joint analyses with a collaborator. So it's really, if I can say it, more like a fee-for-service type of project. You can have kind of a more of an engagement, but again, there's another, before I get to that, the next one is the not engaged one, where the difference is I think that it has to make sure that there's no federal award. I think that's a key difference between the two of them, but they're kind of similar in other aspects of it. The third aspect, the third type of protocol, which most Broad projects are carried under, it's sort of like a project that is of a secondary use type of protocol, where you're not involved in the original project. The samples were collected for another study, and now you want to sort of bring them in for your research because they have a wealth of phenotype data available. And for that, again, a key thing is that there are no linkers that the PI could have directly to the subjects. That's a really key point to this one, and that's kind of an exempt type of protocol. And obviously, now that we do several direct-to-patient type of studies, and those basically definitely require full IRB review, and there's a lot of more scrutiny towards that. But we're totally able to execute those and keep those afloat. I'm not going to go through too much time through this slide, but I just wanted to make sure that I'm sure some of you have heard of this, about the GDPR, which is a General Data Protection Regulation rule that came out of Europe, that was effective in May 2018. And essentially, they basically kind of want to scrutinize further, what happens to their samples, to their data, to the European Union's data, to European samples. And it's a little, the Broad we're just making sure we're adhering to this. And generally, it applies to more projects where one would be providing sort of goods and services back, to the participants, which we do less of. However, just to make sure that we're adhering to all of these things, when we do bring in international samples or data, we do require some additional paperwork now, which essentially it's kind of like a research attestation from your PI, from the sample provider PI, or we do have some model clauses that can go into consent forms. for studies that are just starting up. So I just want to make sure that there is this sort of newer, couple of years old thing that has come through, which created a lot of churn earlier, as you can imagine, since there was a lot of international projects we've been doing. And so we had to sort of backtrack and make sure that we were adhering to all of this. But I can sort of talk about this more in detail with anybody separately if they're interested. And finally, along these lines, again, the Broad obviously has an Office of Research Subject Protection, or OSP. There's a portal that's in place where all of our consent forms are stored, all our protocols are stored, etc. But the key message that I want to give here is that the Broad is themselves, like, we do not have our own IRB. So we don't have a board here that reviews protocols and approves them, etc. But our, the office, the ORSP office, works with us and our PIs to review consents and, give feedback and, to make sure that how far will this go? And then what we do is we actually work with our partnering institutions, MGB, BCH and FARBR, et cetera, to basically use their IRBs of record to obtain approvals for Broad work. So depending on, your prime institute, depending on that, we would work with that particular IRB. So that's something very important to note that, and I think that there has been discussion about whether Broad should have their own IRB or not. And I think there's a lot of pros and cons to that are being discussed. And I think one thing that, we really want to make sure is that, being the Broad and knowing the PIs, they really want to make sure that, no one is sort of, no study is, I think we take a lot of pride in saying that, it was reviewed by, let's say, the partner's IRB and, it was approved by them. So we feel very good about that, as opposed to kind of an internal review that kind of might be sort of scrutinized or judged. So that's on the ethics side. So I wanted to talk about specifically about data use restrictions that is kind of in line with the consents that are obtained for any research study. So getting to that, as we generate data, it is really important to make sure and be aware of, like, what could this data be used for, right? So obviously the study that you are doing, the study that you're consenting patients for, and you have approvals for, that's sort of a non-issue. But as I talked about earlier, you want to be able to make this data available, at large for other folks that might want to do some other secondary studies, using this wealth of data that you've generated. So starting at the top, generally there are sort of two types of overall consents. One of them being sort of biobank cell consent. Like, there are many places globally that are, basically consenting at that style, which are more general. So they're very less restrictive in terms of what you can do with the data. And then there are more sort of disease specific consents. So you want to do a study specifically on cardiovascular disease, and you're really consenting for that. So those are generally a bit more specific. So what happens here is that, once these consents are generated and, the patients are recruited, your study is done, in order to assign, what are the limitations on this data, essentially that same consent form goes back to the local IRB. So not the study PI, but the local IRB where the consent, was devised and approved. And they basically review the consent in light of exactly, the language of the consent. So they can then deem the consent to be, it's very general and it's good for general research use, or it's actually very disease specific. And it can go down to that, for example, it's, good for cancer research, or it may be even as specific as breast cancer. And as you can imagine, over time, we are actually, a lot of us, and even sort of the ethical world, are trying to work with IRBs to try to make these consents less and less restrictive, just because, it's a bit of a shame to generate so much data, and then just because of the restrictiveness of the consent, not to be able to use it for other things. But of course, always adhering to what, the participant has agreed to. And then other things, other categories that can be basically given to these data uses are, can this data be used for methods development or not? Sometimes you will even see things about, using it for only nonprofit types of research, or or for-profit, more commercial uses. And then some of them will even require upfront IRB approval, and they'll make sure that that's stipulated in there. So I want to go to actual data. Once you have your data and we are, ready to sort of disseminate it and share it, something that has changed a lot, in the last few years is really, how do we share data? And as you've probably all heard, and maybe even, seen these kind of slides, we've really changed the model of how we share data. Traditionally, we've always generated data and then we've provided to the researcher. And as you can imagine, right, in this, day and age now, we're generating so much data, so much like that, it's very costly, obviously, in terms of storing it, copying it, moving it. Once I've given the data to one individual, it's really hard to, audit anything, right? And to kind of maybe enforce any securities that are needed around the data. And then each researcher is doing their own thing, sort of silo. And so that model has really been, rethought. And now we're really in a place where we're really trying to bring the researcher to the data. So that's essentially basically, and we can do that obviously on the cloud where, the data lives in a certain place and, we basically give access to it to various researchers depending on, their research goals and their approvals that they have for the data, to data access. And this really, as you can imagine, is like much more, reduces a lot of redundancies and costs and things like that. Much more auditable in terms of who's accessing the data. And then, it's also easier to do sort of, cross analyses between different data sets because they're all living kind of in the same environment. And obviously in terms of tools and things like that, there's like the compute, right? Like it's on the cloud, it's elastic, a lot of things can be shared. So it's really the way to go. And I know that there's, some pains in changing the way we do things, but this is something that we're really, really moving towards and doing for especially the large projects. So I wanted to talk to you a little bit some of the platforms that are underway and that you may have heard of these terms. So as I talked about the traditional way of sharing things, like dbGaP and EGA, sort of, which is the European version of dbGaP, essentially. We used to basically register our studies in dbGaP, and then basically they would sort of disseminate the data, and which would mean that you would have to go to their platform and actually download the data from there. And so many copies, and really really not sort of sustainable in long term in terms of how much data that we're generating now. So some of the newer ways that we're putting data together, as I mentioned, in one place is really, you may have heard of the Anvil of by Data Catalyst, GDC, which is sort of the cancer hub. So it's really, these are platforms, and there are others obviously that are coming on board. These are platforms funded by the different agencies, as I mentioned, and where we're really setting it up so that all the data could live in a certain environment that once the person, the researcher has access to, can basically, use and use the tools on the cloud. And that's something that we're really working on as well, is making sure that all the tools are also available in the same environment to use. And again, as I mentioned, I think that it's clear that there's a lot of work to be done there because a lot of tools, are not on the cloud, and there's been a lot of work towards that. But I think we're making good strides. The one thing that, two key things that I want to make sure that, I mentioned is that regarding of the way we're sharing now, one thing that, it's really, really important is that, whether you can have access to the data or not, it's really still strong like adherence to the data use restrictions, based on what the consents have, what the consents say, and what that, determination is, and which is arbitrated by a data access committee, right? So none of these platforms, they're not able to give access without these permissions. And another thing that I wanted to mention was that, so let's say PI1 has done some studies and has submitted data to the Anvil, let's say, and a second researcher has come in and has approval via dbGaP or other DACs that are being put together. to use that data. It's always a good thing in collaborative world to really, uh, sort of collaborate with a person who has generated the data. I'm not saying to, like, always put them on a publication, but even to let them know that you're going to be using your data in this capacity and things like that. I think it goes a long way and shows kind of good faith of collaborative research. Let's talk about money, which is like a big part of all projects. So, I've put here that there are obviously can be many types of funding sources, right? There can be federal funds. You have various grant mechanisms, private money, which we get a lot of at the Broad as well, and obviously tons of philanthropic money. And depending on your funding source, it's actually very important to always keep in mind what the funding source is, because it does change how you kind of use that money. There might be very strict timeline differences between federal and private money, right? And what kind of spending cadence is required, and whether this money must get spent within this fiscal year or you'll lose it, for example, or there might be other entities which might be more flexible in that sense. Sometimes it's not even about losing it, but you have to be mindful of that, because if you don't use it and you don't have it earmarked, people might realize, well, maybe you don't need all that money for this kind of project. So it's really important to keep that in mind. And then in terms of budgeting for the project, right, you want to really upfront, think about everything that you may need for the project, especially projects that are several years out. I've put a few things in here. Obviously personnel, right, like PIs, analysts, project managers, other researchers, money for analysis, right, in terms of like compute and things like that, and which is also in the personnel time to actually generate data, storage of the data, which is not free, it costs a lot of money to store data, any other materials you might need for your project, travel, for meetings, and obviously 2021, hopefully we'll be able to get doing that again. And then obviously there are indirect costs that, let's not forget those, right, to keep all of our lights on. And the Broad has, a sponsored research team basically that can help, put these budgets together and help apply for grants and things like that. So that's, again, with the help of your project manager, can help you do this and think about everything that should go into your budget. Using specifically money at the Broad, just wanted to mention kind of a couple of ways to do this. You may use Broad as a prime institute. So you're putting in your R01 or your contract to one of the funding agencies as Broad being the prime. So with your Broad hat on, and it goes directly through the Broad, which means all the money would come to the Broad, and you basically have your project done there. You may, and we have many of these, have your affiliate institute as the prime. So, MGH or Brigham, whatnot, that's where your main grant is going out from in terms of application. And then you may want to set up sort of a subcontract to the Broad to do part of the work, whether it's data generation or analysis or things like that. And then we can definitely do this, again, as I mentioned, as a subcontract to the Broad, or even setting up quotes kind of with a purchase order at your prime institute. There's a lot of different ways to, bring money to the Broad and to do work at the Broad. And just to mention that, we actually have moved to the SAP system, which is a more sort of robust and auditable financial system for quoting and things like that. So, which, again, was a transition, but it's much more robust and can be audited, which is really important, obviously, as you can imagine, for finance. Just want to let you know here is that obviously, we've been working with a lot of groups at the Broad to really figure out how can project managers really track projects. And we have still a lot of work to do under that umbrella, and which we're still working on. But, good old Excel is still really heavily used. And then we also use things like Airtable and Smartsheets, which really can be nice snapshots of, where things are for the different projects. You can really lay out by collaborator, by sort of task, by sort of sprints, however your projects are, to really lay out and kind of like, what the kind of progression is. And it's a good way to roll things up to basically show at a meeting or show to your funders or things like that. So we have many of these kind of tools, and we're working on a lot more actually, just to make things more integrated. So I thought I'd take the next few minutes, we're doing good on time, to go over a specific project that I've managed, and to kind of, you know, bring together exactly everything that's been done to basically enable this project. And this one that I just picked is the CCDG project, which is the Centers for Common Disease Genomics. And, you know, again, at the outset, the most important thing is the goal of the project, right? And this is actually directly from the NHGRI's, you know, webpage. It's basically, you know, essentially it's a collaborative, large-scale genome sequencing effort to really understand, you know, identify rare risk and protective variants through many different, you know, multiple common disease phenotypes. But, you know, in that they have like many sort of sub goals, you know, in terms of design of the studies, the architecture of the studies, and also to develop tools and resources that can be applied to sort of multiple researchers, research for the community. And, you know, how the project is organized. You know, we have many stakeholders. So at the Broad specifically, the PIs, you know, Eric, Stacy, Mark, Sake, and Ben are like the key PIs at the Broad. And so they are sort of the key stakeholders to make sure that, you know, they are a vested interest, and making sure that they are, you know, basically given all the updates to. There are other CCG centers that are also generating data, and we're going to be doing a lot of joint analyses on these. So they are also our stakeholders. So, you know, Baylor, you know, WashU in St. Louis, New York Genome Center, Harvard, Rutgers, that is the data coordinating center for CCDG. The sample providers, right? The hundreds of sample providers that, you know, we bring samples from, they are our stakeholders for this project as well. And of course, NHGRI, we know who is our funder. In terms of organization of the consortium, this is something that, you know, Chris talked about too as well. You know, we have monthly steering committee meetings, which is, I think for CCG, we have a big number still. I think there's maybe about 30 people or so on that. And then we have many, you know, other monthly meetings, and some of them are bi-weekly, you know, specific data analysis meetings, data flow meetings. There's a methods meeting. And then you have different disease working group meetings as well. So as data is generating, deciding on sort of like, you know, what kind of analysis we want to do that are disease specific, things like that. And then yearly, we have like a face-to-face meeting with the whole consortium, and then also a yearly teleconference. So the project deliverables, right? So the funding, as I mentioned, was NHGRI, funded, you know, millions of dollars over 5 years to like many centers. And then we have especially, you know, a specific cadence that we have to adhere to in terms of spending, which is aligned with how much data is expected to be produced for every year. So the key deliverables for this project was, you know, over the 5 years, to generate 200,000 exomes and about 140,000 genomes across, excuse me, sort of common disease areas. And 3 sort of overarching areas was cardiovascular, neuropsych, and autoimmune. We also wanted to generate some common controls that can be used, you know, for these analyses and also for other projects. Being, you know, a 5-year project, obviously data is generated over time. So the plan was to generate sort of data freezes over time to allow various interim data analyses. To just let you know, for CCDG, we had, you know, I think after the end of year 2, we had freeze 1, then we had a freeze 2 in year 4, and we are just the final freeze of the program will be its freeze 3 basically. And then the final freeze, you know, that whole data set will be basically analyzed and, you know, basically put through various variant colors across the different centers. You know, like the board will do GATK, and then, you know, there'll be other colors used at other centers. All of the cohorts, so all the samples, all the data, all the cohorts, you know, must be registered in dbGaP. And the sharing platform for CCDG is the ENVIL, which is also NHGRI funded. So that means all of these data points must be submitted there as well. So bringing back what we talked about at the very beginning in terms of the project lifecycle, I want to kind of display how, the CCDG project lifecycle worked. So in terms of the compliance, basically all the centers basically put together secondary use protocols, because that's what this type of project is. We are bringing in, samples that are already collected for other projects at, different places globally, and we're bringing it in for CCDG and for this specific project. So it's kind of a secondary use. Many, MTAs and data use agreements have been, executed. There have been we've used over like 200 unique consent forms across all the disease areas. The funding it's NHGRI funded, hundreds of millions of dollars over 5 years. We have funding allocated towards obviously all of the data production, but also some analyses, personnel, storage, some R&D along the way as well. We've brought in, as you can imagine, hundreds of and thousands of samples from different sample providers. Lots of work gone through to receive the samples, QC them, making sure the naming convention makes sense, and then bringing in all the metadata and the phenotype data, again, which is invaluable to do, any analyses downstream. All of this work was run through GP production. And, obviously the key thing is to really review the data really well, making sure that it's of high quality, very robust data. And then again, delivery back to the sample providers who have an onus on this data, and then to the consortia, right, to be able to analyze this data. And then finally, because again, it is NHGRI funded, we have an obligation to submit this data for the world to use once they have approvals in place. So all of these consents, have to get registered into dbGaP with their use restrictions, so that it's paired with that, so that, when a researcher applies for this data, the data access community can review what the consents have agreed to, and then based on the research aims, give that sample approval or not. And once they get the approval, they can get access to the data, which is also being pushed into the Anvil.
Namrata, before you continue, there's a question about the sort of the broad issue of risk management, and
Yes.
the attendee asks about the scope of risk management, in particular, how specifically it's defined, and what happens when something unanticipated happens.
Yeah, so thank you for asking that question, because I think that I think about risk all the time, and so I think that in terms of risk management, as I mentioned, that there's always things that you can think ahead of time, right? Like you can lay it out, and because it's kind of a known risk, right? And you can sort of plan for that. I think the challenge is when you have the unknown risks, right? Like, so let's take COVID as an example, right? Like that was something that happened where nobody anticipated, and essentially I think that it sort of depends on what the risk is. What is it impacting, right? Is it impacting ability to do the work? Is it impacting ability to spend money? Is it impacting, sort of like the collaboration? And what matters most, right? So the decision that you make in terms of how to mitigate that will sometimes really depend on what that risk is, and deciding on what to do about it, and what those implications are. So it's kind of it's a vague answer, but I think the key thing is to be transparent about those risks, right? So the known risks you can be transparent about, and then as soon as there's something unknown that sort of rises up or bubbles up in a project, I think it's really important to be transparent with all your stakeholders and say, this is what happened, and why it happened, and these are our possible solutions to mitigate it. And that's the best you can do as a project manager, really, right? Is to make sure that to really make sure that, again, being transparent and sort of solution oriented. Now, how can one really avoid as much as possible like getting into situations where things pop up without knowing? I think it's really about having the experience and thinking things through up front and in planning things. and really trying to assess, what could go wrong and trying to see if we can like chart that out from the get-go.
Thank you very much.
And so just to finish off on CCDG, after all of this is done, like kind of what are the next steps, right? So obviously there's a lot of sort of joint analyses across CCDG centers. The disease working groups, span the different centers. So they're working together to think about this. Obviously publications, which many have already happened across, since the last few years, but there will be sort of a flagship paper coming out from the program, and many disease specific papers, and possibly also methods papers and things like that. And then of course, various follow-up studies, but also we're all also thinking a lot about how to collaborate with other programs to further increase power, such as like TopMed and other like Kids First programs. And this is my last slide, and I just want to sort of end by, it's a specific quote from the sort of the guide to the project management, and it's really about lessons learned. And it kind of goes to that question that I had about the risks. Actually, is really, as we do projects over time, something that I really feel that with the experience that we get, the knowledge that we get during a project, we should really like use that, right? Use that to basically address, what went wrong, what could have been done better. And it's really for the purpose of improving the next project, right? And so improving that or improving the next iteration of the same project. So I think this is something that we should always keep in mind in terms of like, as things happen, what could we have done better that we'll make sure we do, next time. So I think those are the kind of things that always kind of keeping in mind. Again, when I'm going to plan the next big consortium project, what things that can I make sure that I need to address upfront, knowing, all the examples and experience I have from previous ones. So that's always a wealth of knowledge to really share. And, that's one that, one last thing I want to mention was that, we actually have a great, network of project managers at the Broad now. And, we really like help each other and use each other's knowledge in order to make things project work, really seamlessly. As an example, if I've never really done, for example, if I'm starting a new project with, a particular funder or a particular group of people that I've never worked with, but I know that, my colleague has worked with them, I will definitely, have a conversation with them and ask them about what things that should I make sure I, worry about, what things I should make sure that I address and things like that. So having that network is really, really helpful, and to learn from others in terms of what lessons they've learned.
Just a question following up on the risk question from a couple of minutes ago. So in terms of a newly identified risk, for example, COVID, is everything in the charter redefined, or is there some other way of communicating and addressing that?
Yeah, so I think that, so if I take CCDG as an example, right, like our year 5 was the COVID year, for example, right? So we didn't redefine the goals, again, because it was in year 5, but we definitely extended the timeline for it, right? So we talked to our funders, we talked to our sample providers, who obviously were also kind of in lockdown situations, so we were not able to get samples from them. We were obviously not able to do the data production in the time that we anticipated, so we spoke to our funders, and as most of the world, like we got extensions to things, right? So in that case, we actually did not need to change the overall goal of the project, but we had to extend the timeline, for example. Now, I'm sure there are examples, and I can't think of one off the top of my head, where I think that if, let's say, you're going towards doing a certain research goal based on a certain technology, let's say, right? And for whatever reason, that technology has evolved or changed, or it just kind of, sunsetted, you might want to change your goal, right? And I think, there's only, you're not going to change your goal in like, towards the end of your project, right? But depending on where things are, you may steer things a bit differently. Again, like if there's a brand new technology and you want to take advantage of it, you know, towards the end, you might start to, you know, request for like sort of supplemental funds, or, you know, repurpose some funds for this sort of new technology. But you can't do that in silo, right? It has to be done with the stakeholders, making sure that there's an agreement in place. You may want to, you know, even go back and kind of revise if there's like a set statement of work put through with your collaboration, you may want to make an amendment to that, and that can happen, absolutely. But I think the key thing here is that not to go kind of rogue and do things on your own, right? It has to be really done in a capacity where everyone is made aware and agrees to the changes.
Sounds great. Thank you so much, Namrata. That was really, really helpful. And I look forward to the next talks in this series as well. So thanks very much.
Great. Thank you, everyone.